aim so high. I don't think anybody I work with is that much taller than me, except for Dr. Rose. Okay. Finally. You guys are probably gonna get sick of me complaining about this computer. I have to like set up all of like the actual programs and like re-sign in to like even use like PowerPoint, and like I get dual pushes for everything I open. So you're gonna get sick of me complaining about it, because I will complain about it pretty regularly. Because I think it's just nuts that I have to do that every time. It's just stupid. So, all right. And like whenever I'll be done with this today, I have a class in the evening, I'll have to redo it again. So it's the stupidest. That's all I can say. All right, let's get this going. We're gonna do a Zoom activity. Not Zoom, sorry, excuse me, Kahoot activity. I have chapters 1 and 3. Their Kahoot quizzes are separated, just FYI. So we're gonna go through today in class, we're gonna play chapter 3. So if you want to go over chapter 1, those Kahoots are posted on the modules area. So you're welcome to go. You can do these as much as you want. This is never going to be for credits. This is all for like just review. And I like to just review the topic we went over last time, so we can remember that we learned a thing last time. You can put in when you guys are joining, and I'm about to start it. When you guys join this, you can go, if you're using the website, kahoot.it is where you will go, okay? And then you'll enter in that number at the top, and that will let you enter this thing, and you can enter whatever nickname you want. It doesn't have to be anything that identifies you, because if you think you're gonna do bad and you don't want people to know, just, put, dinosaur or something. Then if you're using the app, you should be able to just open it and then enter the pin in there. And you can always just use the QR code. This room's so big, it's kind of hard to see it, but yeah. You're welcome to come up closer if you need to do that. So we will do one of these to review the previous chapter every morning. Like we'll come into class and that'll be the first thing we do. So the next class we'll go over chapter 2. Let's try to push too hard on this. Anybody have any questions about the content or homework or anything like that? Yes. Yes. Yes, I know a lot of you are coming from chemistry and they had you print out the entire world. We are not doing that in my class. That's a waste of paper. So all of my stuff is online as much as I can. And when you guys come to my lab and do my lab exercises, and we'll walk through this when we have Tuesday's lab, okay? But in my lab, it's going to be the worksheet that's associated with it. And you can see that as the PDF that's on top hat associated with each exercise. You're welcome to print it out if you want to. However, I will provide a copy for each person in one of those dry erase envelope things. And then you'll answer the questions with that and take a picture of it, upload it on top hat, and then erase it. And the next person can use it. So we're not wasting as much paper, and all you have to worry about is making sure your dry erase marker works. Okay. So we'll have that. And like I said, we'll go over that on Tuesday. Tuesday's lab doesn't have a pre-lab. If you've noticed that, like it's like the pre-lab that you see first is for the 28th, and we have lab on the 26th or whatever. I guess you guys are the 29th and the 26th. Yeah, the day after. But anyways, those we don't have a pre-lab for Tuesday, so don't worry about that one. We'll kind of talk about what the pre-labs are for and everything like that when we get in there. We do have Kahoots in the lab as well. They are going to be different. So we'll have these Kahoots going over the previous thing. The lab Kahoots are going to be going over the thing we're doing that day, because supposedly you read the lab manual and you answered the questions and you came prepared to lab. And so we'll be doing, it's just 5 questions Kahoot before we get started to kind of review what we should know. Then we'll give the real lecture about it. And then we'll actually do the experiment, and then you'll fill out your little worksheet and take a picture and post it. And that's pretty much what you do for lab. Okay. My lab stuff is largely participation based. The only thing that you can get points off on are the pre-lab stuff. So do be sure that, you're looking up your answers and make sure you're getting them right. And then the quizzes after the lab, use your notes, man. Like it is timed, but that's just to make sure that you guys are like actually in there engaging with it. It's plenty of time, I promise you. Sort of like, have any of you guys done any of the chapter quizzes yet? Anybody? It's okay. They're not due till before the exam, so it's all right. But if you were to want to do them as we go through the chapters, you would find that it's 10 questions, multiple choice, and those are 15 minutes timed. It's not in lockdown browser or anything. So you can use your notes, you can talk with people, you can work on it together if you want to, I don't care. But again, the timer is there just to make sure that you are actually sitting there focusing and doing the thing as opposed to just milling around and BSing and not putting effort into it. So and be sure you get it done, because I have so many people that'll start a thing and then they'll email me and be like, I got busy and I didn't finish it. And I'm like, yeah, it looks like it. How is that going to work at your job? Like, is that going to go well for you? So, yeah. But when you guys get to the quizzes on the lab stuff or on the lecture stuff, that's what you can expect, okay? Use your notes, it's totally fine. Work together, that's totally fine. I don't care. Just be sure you get them done. All of those are going to be due before their exam. So, for example, chapters 1 basically through 7, minus number 6, are going to be Unit 1 exam. So all of the quizzes associated with those chapters are due right before the Unit 1 exam. Does that mean I recommend waiting until then to do them? Absolutely not. They're there for you guys to work at your pace. If you put them off until the last minute, you're going to be hard pressed to actually get any actual studying done, because you're going to be worrying so much about your grade for your quizzes. Okay, so don't let them sneak up on you. So just so that we're clear, you have quizzes for every chapter and you have to get them done by the exam date. Do them on your time, knock them out on weekends if you want to, but do make sure you get them taken care of. That way when studying comes around, you can actually study and not worry about quizzes. Yeah. Remember, we have a day before each exam, a class period before each exam that we're putting aside for the unit projects. So that's the group projects. So on that day, literally all 6 groups will come in front and basically be like, hey, this is our poster. It's explaining how, this is how germ theory works, and Pasteur did these experiments, do da da da. I expect kind of all to work together, the 4 of you. Probably most of you have 4 people in the group. But so you have 3, that's okay. But for you guys to work together kind of to present it to the class. That's all it is, just reviewing that topic. I think I've said before, but you don't need to go crazy on it. I don't need you pulling out resources from like the CDC and like all this stuff. If we aren't having to learn that much of it in the class, I don't need you to go over that much of it in detail. One of my biggest pet peeves, I used to have these extra credit assignments where people would write either summaries of each chapter and get like a point for that, or write questions based on just summaries of the labs and questions based on the chapters. But one of my pet peeves was I could tell that some people were just like asking ChatGPT, write 3 questions about, genetics, and then popping it in. And I was getting points for it because I can't, you guys probably know this, I can't actually prove that you're using AI. And it's my bad for not making it clear that, I was going to get mad about it. So it's whatever. I used to just give people the points for it and not argue about it. But I've eliminated those because of this. Okay. If you're using AI and it's helping you and you're understanding what the AI is telling you, that's one thing, right? But if you're just typing it in and then copy and pasting and you don't even know what it's giving you, and you can't, if you read it and you don't know what it's saying, you've got a problem. And it's your problem, not mine. Because if you're just copy pasting things, how are you learning anything? The whole point of most of this crap is just so you guys will learn. I'm not getting anything out of your assignments. I don't care about your assignments. You guys should be doing it as tools to help you learn. So if you start cheating on the assignments like that, that is not a penalty for me, just FYI. Okay. And I can tell sometimes, not always. If you're giving me a lot of questions that ask questions about, chemical reactions of metabolism that we never talked about in lecture, yeah, I'm going to be looking at you. Because you need to be writing questions about what we're learning, what you need to know, not just pulling stuff out of your butts and that. So I recommend the same thing for the reviews. Like I said, I'm not going to be super picky about it. But if you want to do some AI to help you find wording and things like that, as long as you understand what you're putting on your project, it's fine. As long as you understand it, okay? I think I have that like clause in my syllabus about AI, you know, if I explicitly tell you you cannot use it, you may not, but other than that, I don't really mind it. So I'm telling you guys, if you want to use it on your projects, you can. Just be sure you understand it, because when you guys are presenting your projects to me, if you're talking about term theory and ChatGPT wrote the whole thing and you think I'm never going to know because you double check to make sure it covered everything. If I ask you a question about your project and you don't know what I'm talking about, who's in trouble then? So be aware of that, because I can call you out if I choose to. So know the stuff that you're putting on your poster or whatever. That just gives you more incentive to make sure that you're only focusing on what you need to and not putting extra crap on there. Okay. So yeah, all you have to do is summarize that topic. It sounds simple enough, but people overthink it. That is usually my problem. It's not that people aren't smart or something. It's that they overthink things. You know, when I was asking you guys the questions last time in class, not everybody stand up, I'm going to do it today. But when you guys get called on, or like even whenever you weren't called on, but you're trying to think about that thing, how many of you guys were overthinking the question? Probably all of you, yes. So that's usually the biggest hurdle that my students have to overcome. It's probably not as complicated as you think it is. Take the time to read the question and understand it, and think about what I told you. If I wasn't talking about all these complicated things, then probably not asking about all the complicated things either. So we'll do that today. Think about that whenever you get called on. If you guys haven't noticed, on the front page of Canvas, on the home page, like almost nobody ever visits, if you scroll down to the bottom under that little orange banner thing, I have each section's like scores on their points for the group questions. Right now, some of them are, you know, weighted towards groups. I just happened to pick a whole bunch of Delta. And so if there's a whole bunch of points for Delta, it doesn't mean that Delta is necessarily better. They just got more questions so far. So just hang in there. You can see that. That'll eventually even out. But yeah, makes sense? So Tuesday, big thing, so I don't forget to tell you at the end. Tuesday, big thing is we will have lab after class. I tell all of my classes the same thing, which is if, you know, we are lucky enough, in these first few days, I usually take up time because I have a lot to tell you guys, but if we're lucky enough that our lectures finish out early, we're just gonna go straight to lab, all right? That way we finish lab early. That's the goal. That I'm, you know, lucky in that I'm the only one that runs micro, so I'm only using the lab, so I don't have to wait on anybody to get out of there or anything. So it's always just going to be my lab and my class going into that lab. So we'll just do that. I've had a couple people say, and I even had just today a couple people who have some issues going on where like maybe they get stuck at the hospital for a loved one, or they are sick, or whatever it is. Remember, you guys, get your doctor's notes. Just go to the freaking, you know, urgent care. And they're like, yeah, you're sick. Have them write you a note. Like, I don't even need you to get a prescription filled if you don't want. Like, just have somebody verify that you were out for a reason. If you have a loved one that's in a hospital, you can ask those doctors just to write a note saying, yes, this person was here with their loved one. You don't want any details about your loved one or anything. Just have something for me to prove that you were out for a reason. Okay? If you do that, most of the time I'll let you make up your points. Almost always. People who are missing lecture today, you guys know that you get attendance points. If you're joining on Zoom, you automatically get your points just for being on Zoom, because that's the same as attending, in my opinion. But if you can't join on Zoom because maybe, you know, if you're at a hospital and somebody's sick and you don't want to be on Zoom or being inconsiderate or whatever, I understand that. So what I am having people do is basically they will watch the lecture. And again, I'm not monitoring that, right? But they will find one of the questions that got asked in class that, you know, I have you guys answer, and they will tell me the question, tell me the answer, the correct answer, and tell me why the correct answer is the correct answer. And as long as I do that with at least one question, because I only ask each of you one question typically, if you even get called on, then I give them points for attendance. Fair, right? So FYI, if you have something like that happen, you can earn those points back. You can earn points back for labs typically. Just be aware that you can't earn the absence back. So I can't do that. That's against policy for the science department. So you can get the points. And if you're only going to miss a couple labs here or there because you're sick or because, you know, you couldn't get daycare for your kid or whatever it is, then that's usually fine. But if you guys are, you know, going to be missing a lot, remember you can't miss more than 5. Okay. All right, let's do this. So there's at least one question here that doesn't apply to you guys anymore. I forgot to update it, but I'll let you guys know what it is. All right. If you guys do this online, it'll just be by yourself. You can take your time with it. All right, so which of the following is not one of the 5 eyes? Okay, all 5 of them are up here, only one of them. So that's, you guys did great. That is correct. So this is going to be infection. We know you're going to have, inoculation. You're going to have whatever your throat swab is and inoculate your media, introduce the bacteria onto the media. You're going to incubate it so it'll grow. Then you can go forth with isolating the thing that you're interested in. And then you'll be doing your inspection, which is doing tests with it to get answers, and then identification based on those results. So that's the 5 eyes. They're in order for a reason. I keep forgetting that my screen is not the same as what's up here. Fixing out. Differential media. It's going to ask about differential media and then selective media and then enriched. All right, so most of you guys are getting this one. And it says differences in there, so that's part of the giveaway on this one. So differential media tells you the difference between things. So it's going to highlight biochemical or metabolic differences. And yes, we might be thinking, okay, that's the one with the color change. But this is where I'm saying, like, be sure you can think differential means color change, but why is it changing color? Because of products made from certain metabolic pathways that some have and some don't. Okay. This guy makes acid and this guy doesn't. Next one is, I believe, selective. So that you guys are prepared for what's happening. All right, selective media. This was less time. Okay, so majority of us got this one. So with selective media, our little hint is that selective media selects for the thing that you're interested in and the other things won't grow. Obviously, none of these choices say select. And this is exactly what I'm talking about when you guys are looking at your study sheet. Don't depend on word for word. You better know what that means when it says that. So when something is selecting for one thing and not the other thing, that means it is inhibiting the growth of some organisms and encouraging the growth of others. So that's what that's saying. It's the same exact thing. It's just a different wording. Be sure that you understand those statements well enough that you could reword them with different words and still understand them, because that's how the test is going to be. It's not going to favor memorizing words. Okay. What type of media supports fastidious bacteria? I kind of already gave this one away. I forgot that it was this way. You guys know what I said. I said it was going to be differential and then selective and then this other thing. This is the answer. All right, so it's going to be enriched. So if we're talking about complex, remember complex, it just means that there's an ingredient in there that you don't know the exact components of. That could include something like a growth factor, which is what we're talking about with enriched media. But you also could have that with defined media too. So That's kind of the opposite of the complex. It's other side, right? Enriched just means you have that growth factor that fastidious organisms need, like our vitamins. All right, streak plate techniques. What do we use that for? It's not what they look like. Okay, so streak plate technique. There's several different ways that we can isolate microorganisms. Really the only one that I'm expecting you guys to understand is the streak plate technique, because it's the one we're going to be doing in lab. So we're only going to focus on that one. And it is the most widely used. I mean, I've used it in all of my jobs pretty much. So you can also dilute. I've used dilutions too, but we're not going to worry about that here. So which microscope has a higher resolution? This is one we didn't go over, but make a guess for the fun of it, I guess. What do you think the answer would be? The resolution is like you can magnify it the most. Smaller things. Just so we're clear. And I don't expect you guys to know this for the test. It's just one I forgot to take out. So that's electron microscopes. And they just shoot electrons at things and then generate an image. So you can see down to atoms with those. Yeah. So it's one of those things that, like, I used to teach it in this class. We're never going to see one of those in our class, so I don't care that much about it. So this is a light microscope. Name the part that's labeled. So that's the thing where that line's coming out. It's hard to see it with this big room. We did go over those parts. Talk about all the parts. We did talk about those. That's the objective lens. Good job, guys. So that's the one that's going to see the specimen first, creates the real image, right? Remember, ocular lens is what you look through, and it creates the virtual image. So stains improve contrast. Is that true or false? Contrast is, like I said, black on white as opposed to light gray on lighter gray. So this is absolutely true. This was kind of a gimme, if you will. That's the whole reason that we use most of them. When we do a simple stain, it's literally the reason why we're doing it. All right, gram staining procedure. We did not go over this in detail. We are going to be expected to know it in chapter 4 and in the lab. So give me a guess. What of these do you think would be the mordant? And I'll tell you, a mordant is just like brick and mortar. So it holds things together. Let's see if you guys can guess. All right, so this one, like I said, we didn't really go over this in detail, but you will have to know eventually. In the gram stain, we're going to put crystal violet. It's going to get stuck in the peptidoglycan that we're going to go over in chapter 4. Then we put iodine in order to lock everything in place if it got stuck there. Okay. Then we rinse it with alcohol to remove anything that didn't stick. And then we counter stain with safranin, because if we just rinsed out all of the purple because it didn't stick, because you didn't have a lot of peptidoglycan, then now you have clear cells again. So you can't see those very well. So we add the safranin for contrast. Okay, so that's what it would be. We'll talk about which one's positive and negative and what that means in chapter 4, which will be the next on Tuesday. All right, acid-fast staining. What do we use that to diagnose? I forgot this one. Again, details in chapter 4 and in the lab. We're going to be learning this in the lab. We're going to be doing this in the lab. But they are useful for diagnosing specific things. Just so you guys know this. If you guys ever worked in a hospital and ever seen an order for AFB, that even if you didn't order it, you just saw it in, like, I don't know, Epic or whatever. So AFB is acid-fast bacilli. And acid-fast is the stain that we're going to be doing to identify Mycobacterium species. That's the only species that I need you to care about when it comes to acid-fast. All right, it's important. Let's say, so first of all, let me warn you guys about this, because a lot of people think that tuberculosis is like this old world disease that whatever, who cares about it? I mean, you can care about it, I guess, in third world countries, but if you don't really have it here, you're wrong. 1/3 of the world is infected with tuberculosis right now. That should be shocking to you, especially when we're talking about things like consumption. Maybe you've only seen like Moulin Rouge, and that's like what you think about. when you think of tuberculosis, but you're wrong. It's rampant in the homeless population. So is HIV. So it shouldn't be surprising that they're getting these sorts of diseases and harboring them. I'm gonna tell you the reason why it's so prevalent here in a minute too. But the first thing I want to do is tell you guys that a couple years ago at one of the Edmond high schools, 14 people were diagnosed with tuberculosis. Healthy people just like you guys. There's one faculty and the rest were students, high school students. So, I mean, most of the time you can carry TB without symptoms. So that's why they have you guys do the PPD skin tests. When you go to work at a hospital or healthcare facility, they usually have you get those, like, you know, pricking your skin. They come and see if it's swollen in like 2 or 3 days. Yeah. And we'll talk about how that works later on in the immunology chapters. But that's looking to see if you've been exposed to tuberculosis. If it swells up, then you have been, or you've had the vaccine, which they don't give here in the United States. So yeah, it's common enough that hospitals think they need to test everybody that comes in there to work for them. So think about that. It's just we don't talk about it a lot because it's usually poor people that have it. That shouldn't surprise you either. So thing I was going to say, it's rampant in the homeless population, and the reason why it's rampant in poor populations or in third world countries, when you treat tuberculosis, and it is treatable, you can cure it, there's antibiotics for it. They usually start you off for your first 2 months, y'all, of your antibiotic treatments with 3 different antibiotics. The one that you respond to the best, they will stick you on for the rest of your 7 months after that. And you are the one who's responsible for taking those pills. Ever gone with like a UTI, your doctor's like, take all 10 of these pills, take all 10 of these pills? Well, if you don't take it all the way through and kill off the bacteria completely, if you just stop taking your antibiotics when you don't have symptoms anymore, the reason that doctors are getting mad at you isn't because they're just jerks. It's because you're probably, contributing to antibiotic resistance. And if you don't think that's a problem, what do you think is going to happen when we cannot treat TB anymore? And some people think like, well, that's not going to happen in our life. And we're not going to see bacteria that can't be treated. What do you think MRSA is going towards? Yeah? MRSA, methicillin resistant. So methicillin is a pretty staunch antibiotic. So it's already resistant to methicillin. And we can go vancomycin, and we can go to stronger antibiotics, carbapenems and stuff. But once you do, they're more dangerous for the patient. So if you've got somebody who has MRSA, who already has kidney disease because they have diabetes and they don't have good blood flow, they stubbed their toe, they got MRSA and whatever. That's all it takes. And then now, how are you going to end up treating them? You can't treat them with vancomycin or carbapenems because their kidneys are failing and they can't process it. What do you do for people like that? You guys want to know what you do for people like that? You let them die. Yeah, comfort care. That's exactly right. We do that. Now we have an issue with this right now with gonorrhea of all things. Around the world, there have been strains of gonorrhea that are completely antibiotic resistant now. We have none left. So what do we do to these folks that have gonorrhea that's completely antibiotic resistant? They're not dying, right? They don't need comfort care really, but they are sent home and they're told, if you're going to have sex, please, use a condom and all that. How do you think they got the gonorrhea? Right. So yeah, that's what we're dealing with right now, guys. And bacteria can communicate with one another basically and trade these resistances with each other. So that should be terrifying. Is a reason why it's terrifying. It's also the reason why we need to lean more towards non-antibiotic treatments, other things that maybe the bacteria can't get resistant to. So that's in the works. Okay. Oh yeah, here's your podium. I know you guys were waiting on this. And we definitely have time to do chapter 2. I just don't. It might be another one of those situations where, as I get excited about talking about tuberculosis or whatever, that we end up not having to do the last questions, which is fine. I'm sure you guys won't cry about that. All right, we ready for the real deal? Congratulations on you guys. Let's actually do this boring talk when I find my cursor. All right, so chapter 2, we're going to be reviewing chemistry. There's gonna be stuff on this where I'm gonna talk about it, but I'll run through it, but I'm gonna expect that you guys have heard it before and understand it to some degree, okay? So most of the stuff you'll recognize, not kidding. but I just want to be sure you guys understand it well enough that when we get into it in the terms of microbiology, because I hate to break it to you, maybe you thought you were getting away from pH. Nope. We use a lot of pH. It's extremely important. And hydrogen bonds, probably the absolute number one most important thing in microbiology, in our course anyways, is hydrogen bonds, which we'll talk about. No worries, I'll explain to you in my terms. But you guys know what an atom is. Proton and neutron in the middle and the nucleus, and electrons floating on the outside. What charge do electrons have? Negative, right? Remember that because that's important. Here's an atom. Here's a periodic table of elements, okay? Yeah, this is how this is gonna go. All right, molecules, that would be any time you have two things bound together. Oxygen, O2, that's two oxygens together, it's a molecule. H2O, we've now introduced an H with that oxygen, right? So we've got two different kinds of atoms together. That's gonna be a compound. Is it a molecule also? Yeah, so it's both. But if you only have one kind of atom, that's only molecule, it's not a compound. I'm probably never gonna discern between the two. All right, so the three major types of bonds that we need to understand for microbiology: covalent, ionic, hydrogen. Those sound familiar? Because they should. Covalent bonds. You should know at least the basics about these ones because I will absolutely ask about these on the test. And it will be sort of something along the lines of what type of bonds do you see sharing of electrons? Or what type of bonds do you have donating or transfer of electrons? Because those are the two that we have electrons going on with, okay? So covalent bonds, these guys are sharing their electrons, okay? Think of it as you are playing ball with your sibling and you're just passing it back and forth. You're sharing it. If we have one electron, just sharing it, okay? That's a covalent bond. So covalent, I think of two, and that's gonna be sharing. They're sharing equally. All right, let's say that my sister and I'm the older sister, and my sister's younger than I am. And I was a spoiled brat, okay? And we might play ball together or Barbies or whatever it is. We might have the Barbie that we like, and I'm playing with it and she's playing with it, but I'm hogging it more, yeah? So I'm the one that has the Barbie the most. Now imagine that that's an electron. So if I'm hogging that electron, that Barbie, more, then that would make me have a polarity of negative. We haven't separated anybody apart. They're still sharing the Barbie, they're still sharing the electron, but one of them's keeping it more often. So it tends to have that negative charge to it, that polarity, okay? But we're keeping that covalent bond there. When does this happen? Absolutely seriously important with water. I know it has a picture, but I'm going to be drawing something big that's going to be crazy. So if we have water molecule, this sucks when I do recordings of this because it can't hear me when I talk. And it also can't see what I'm drawing, but I have to do it. So we have these charges, right? Oxygen is electronegative because it's keeping the Barbie more. It's keeping the electron, which is negatively charged. Right? So that's why you see that. So this is a negative polarity. This is a positive polarity. Understanding why this happens is key to understanding the hydrogen bond, which we're going to get to. And don't worry, I'll draw all of that too. All right, so that's the covalent bond. It's in this picture. If you guys are watching online, if anybody's watching online, yes, we have somebody. Sorry, I just saw this. I'm so sorry. Sorry. I hope that you saw that number on the screen. That was the pin. I don't know if you got to play. I hope at least you got to watch it, but it doesn't share the same screen. That's a problem. Zoom will like share the screen that I have and not the screen that's up there. It's different, in case you guys don't know. I don't know why, but it is. So we got somebody watching online that was asking, I'm sorry, it doesn't chime when somebody chats with me. All right, next, ionic bonds. Why? There it goes. Ionic bonds. You're going to create ions, go figure. Well, an ion happens when you have this same thing happening. But this time, you might have sodium and chloride. It's a common one, everybody knows. Right? So this would be my sister, and this is me. In the example, okay? So if we were supposed to be sharing the Barbie, then I just take the Barbie and leave to go play with it on my own. I've stolen that electron. So what happens? She is without Barbie now. She's lost an electron completely. So that what we would have had her having that polarity of positive because I was hogging it. Now she's stuck with that permanently. So that's what an ionic bond is. We have, instead of having your electron mostly hogged by your amine old sister, now it's been stolen by your amine old sister. And so now you are without an electron. That gives you that positive charge. This is a true charge. This is not a polarity. Polarity is the direction you'd be leaning, but you're still maintaining the sharing. Not in ionic bonds. You're not sharing. This is donating. She donated her Barbie, right? And this one's negative because I stole the Barbie. I have an extra Barbie. I have an extra electron, so I'm negatively charged. We're not sharing this one. So this is what we say donating or transfer. Transfer is probably better. Transfer, steal, donate, all the same. Once you get the concept that that electron is no longer with sodium and is now with chlorine. And the reason that they have the charges is because sodium lost its electron, which makes it go positive, just like we saw with the thing up here, so now it's permanent. Make sense? Okay. Because it's like an extreme of that. You just kept it instead of sharing it. So you often see with things like sodium chloride, when you put it in water, not surprisingly, because the sodium has an actual positive charge, and the chlorine has an actual negative charge, that, hey, this is negative. This will associate with your sodium, right? And then that will separate it out. This one will associate with your chlorine. Because opposites attract in chemistry. Okay. So that's associations. But that's what causes salts to dissolve. This is the whole concept behind why some things mix well in water and why some things don't. These associations here. And I'll show you why, because this obviously is extreme. Yes, we've got an actual positive atom out here. But what happens if we had our water molecule, and we know why we got a little positives out here, we got a little negative out here, because we're sharing, but this is hogging, this is electronegative. Electronegative, it's got a negative, it's got a negative action because it's hogging. Not nice, not so good. So we got that going on. What happens in water, though? Because it's not just one molecule, right? There's a whole bunch of H2Os in there interacting with one another. And so I'm sure you guys know that electronegative, polar, negative oxygen will be attracted to the positive of the other water molecules. Cool? So what kind of bond is that? You guys know? The hydrogen bond. Okay? Hydrogen bonds occur when you have a polar hydrogen associating with something that's a negative. It doesn't matter what. It's not sharing any electrons or anything. They're only brought together by attraction. Okay? This is a hydrogen. So that hydrogen bond is a weak bond. We're not sharing any electrons. It's only based on attraction. You can see how that would be a weak bond. Because what happens if we're looking at, we're going to get into all of this all at once. So I feel like telling a story with the first time makes it make more sense than we get into the details. But you have this balance of all of this going on. And that's nice and it's happy and everybody's getting along and it's fair. Because things can get a little, because Schrodinger's cat, let's put it that way, okay? You guys know Schrodinger's cat, where it's alive or dead at any given moment until you open the box and you see if it is alive in that moment or dead in that moment. That's what Schrodinger's box is. How do you know, though, until you look in the box? That's the whole problem. You don't know. How could you? So that's kind of like what water does. It is either either H2O, which is nice and fancy. We keep drawing it, and it was, ideal world, it would be like this, except the water, because of its polarities, and because it's not always just fogging, sometimes that oxygen gets nasty and it seals. So when that happens, we have hydrogen ions. That's called hydronium. Okay? But hydrogen ions and hydroxyl ions. This is just whenever this guy gets calorie. So you can kind of see how those can go back and forth. That's what I mean about Schrodinger's cat, okay? So you can go back and forth from this to this. I think what would chemistry do, at least these people like this, they have special symbols for crap. They would say this. It can go back and forth at any given moment. When you have equal amounts of H plus and OH minus, hydrogen and hydroxyl ions, we have equal amounts of those, then it's plain old water. They're equal in amount. You know what we call that when it's equal amounts of H pluses and OHs in your water? We call that neutral. And that is pH of 7. Okay? So when our scale, if you guys don't remember, is 0 to 14. Right? The dead center is 7. So when your H's and your OH's are equal, when everything's at equilibrium, that's your 7. And that's why that's the neutral. Right? We'll say equilibrium because I like that word. Okay. Cool. But then what happens when it's not equal? Are there situations where you might have the H pluses going higher than the OHs? Absolutely. When does that happen? Acids. So you guys know them like the most obvious acid, hydrochloric acid. Now, who cares about chloride? Nobody does ever. So here we have hydrochloric acid. And just like we already talked about over here with salts, it does the same thing. We're going to have chlorine stealing that electron from the hydrogen. When you dilute this in water, you're basically introducing hydrogen ions. These two, H plus and OH, in water is all we care about with pH. Nobody gives a crap about this chlorine in pH. Okay? It is a measure of your hydrogen ions or your hydroxyl ions. We're not going to look at it that way ever in my class. Okay? Hydrogen ions is what we care about. The H pluses, the acid, and how much H pluses there are in relation to what equilibrium should be. Right? So if you have more on your scale, let's just draw it out, right? So here's your 7. This is neutral. H plus equals OH, negative. I put the negative at the O so you guys can remember that that's the negative part of it. But yeah, that's where it's totally like that. So cool, they're equal. As you go this way, have an increase in H plus compared to OH. As you go this way, you have a decrease in H plus compared to the OH. Now the OH is taken over. 7 is where the neutrality is. So people, your pH is somewhere around, I think it's like 7.24 or something like that. It's very tightly regulated for a reason. We'll why that is when we learn about enzymes and stuff, but cool. So here's that concept of that pH scale. It's easy enough just to look at the scale and say, okay, she's telling me that these numbers mean this and these numbers mean that. What do the numbers mean? That's the part everybody hates. So the numbers, what they mean, you have pH, which I want to point something out. Some people I feel like get lost on or confused on for some reason. Why is it called pH? Because we're looking at the H. Okay? Anytime you get confused about what we're talking about, the pH is looking at the hydrogen concentration. Okay, it says it in the pH. Right? H equals negative log of concentration of hydrogen ions. We already knew it was going to be hydrogen ions. Concentration is just how much it is compared to everything else in there. So we're just looking at our hydrogen ions. Negative log is the part that tells us this number here. It's not as complicated as it looks, and it shouldn't be anywhere near as complicated as probably chemistry. So, because all it means, log, Newton means just 10. So 10. Start with that. What does negative log mean? That means that whatever this concentration is, you're going to put a negative in front of it. And because it's log, and we know how this has to be 10 to the negative, whatever your concentration is. Okay? So let's look at it. 10 to, I hate this because I put right out with zeros. Okay, 10 to the negative 7 is our neutral. That means 0.1, 2, 3, 4, 5, 6, 7. Yes. I have this, whatever, but you guys get the idea. Wow, that's a point there. It's a really small number. That's the concentration of the hydrogen ions in that solution at that point. Okay. That's what that number means. It's 10 to the negative of pH 7. Okay, so if it's pH 7, 10 to the negative 7. If it's pH of 2, which is what hydrochloric acid is, just straight up. So that would be 0.01. Look at the difference. That's why 2 is more hydrogen ions. Make sense? That's why all we do is go up to 0, because 10 to the 0 is 1. That's the most you can have. But yeah, so that's the idea of how that's going to be broken down. You get to bigger numbers, obviously this is going to shift way out farther. That's why it's numbered the way that it is. So think of it as, we've got our water. It's supposed to be H plus and OH at equilibrium. When it is at equilibrium, that's 10 to the negative 7, pH of 7. If we're going to acids add hydrogen ions. Most of us know that pH 2 is acid and then pH 10 is base or alkaline. If you can remember at least that pH 2 is acid, then you know that above 7 has to be base. So 7 is neutral. So whatever, so this is base or alkaline. We have our water breaking down into H plus and OH. 7, they're equal. And if you add in more H's becomes acid. That's because this number is shifting to bigger numbers, which means like 10 to the negative 1, 10 to the negative 2. That's going to yield more concentration. Okay. Good? So if I ask you guys on the exam, you have a solution that is a pH of 8, and then another one that's a pH of 10. Which one has the higher hydrogen concentration? 8. Yes. That makes sense here. While we're talking about it, while we're working through it, and while it's written on the board, and you're probably thinking this makes sense, make sure that sticks with you when you get to the exam. Okay? Because there will be a question like that. The concentration one is usually the one that trips people up, as opposed to like everybody can tell me like pH of 8 is acid, but what has the higher, when you phrase it that way, a lot of people get lost in that. So that's the same. All right. Acid just means more hydrogen ions. Cool. So we're about to like click through most of these slides because I did that. We did this. We mix it in water because it associates with the charges. Hydrogen bonds. We already talked about how if that O is hogging the Barbie and the H doesn't get it as much, that H is slightly positive, and it can associate with anything negative out there, whether it's just polar or is a complete negative ion. It doesn't matter. That's a hydrogen bond. They're weak bonds. We have no exchange of electrons going on. It is just based on charge attraction. And the reason it's a hydrogen bond is because there's a hydrogen involved. More important than you think. When we talk about those hydrogen bonds and those weak ones, is what I was trying to get to with pH. You can imagine that if this is your situation, situation with your water molecules, if you're increasing your H's or decreasing your H's, that's going to disturb the ability of this water molecule to interact with that water molecule. Yep, because the H's can come in and just on their own interact with this. Now this guy can't interact with it anymore. That's fine, we're just talking about water. But what if I'm talking about your proteins in your body? Now, your proteins and their structure is massively reliant on hydrogen bonds. Let's talk about DNA, because it's the easier one to draw the picture of and explain. Oh, okay. I'm trying to figure out where to write this. This always happens. I just be going there before, but I think everybody can see it as well. DNA, you know how it's shaped. It's a double helix, and you've got these ladder rungs across. And one of those ladder rungs it represents. So if you stretch it out, there's two strands, right? This one might be a G, and this one might be an A, and this one might be a C, and that one might be a T on that one side of your ladder. And then on the other side, see, I know you guys already learned this, but I just want to lay it out in a way that you can relate it to what I just talked about. We'll talk about A to T and G to C in another chapter, in chapter 6. You don't have to worry about that right now. Okay, but it's just my example for you guys. All right, so this is your DNA strand. One side of the strand and the other side of the strand. This is why I say double helix, because we've got two strands basically going along. One's complementary to the other because of how A binds with T and G binds with C. We'll talk about what that is later, like I said, but this is the makeup of your DNA. How are these two strands attached to one another? Why are they together as a ladder? Why do they make up the shape that they do? Why? Hydrogen bonds. So polarity of these molecules, the G has a polarity that associates best with the C polarity, and same thing with the A and the T, and their shapes and how they interact with one another. So those hydrogen bonds that form because of the polarity, that's slightly negative of the hydrogen, slightly positive of the hydrogen, slightly negative of the oxygen, usually oxygen, right? So it can also be nitrogen as well, but it's mostly going to be oxygen. That's doing that super electronegative, right? It's like, so it's going to steal those Barbies and use it. It's hogging the Barbies a lot in one side, and so because of that, it's going to have positives that will associate with the negatives on the other side, right? That's the whole concept of what's holding your DNA together. Here for all of it. Both strands on all of your little X's in your chromosomes are being held together by hydrogen bonds. It's totally they're weak bonds. They're just associations based on polarity. So what happens if you introduce extra H's into here? When you're supposed to have a 7.24 in your body, and you have more H's than you're supposed to, or even less H's than you're supposed to, these hydrogen bonds aren't going to form the same way, because now we're snaking in, like we did with that water molecule, those H pluses are interfering. The hydrogen bonds aren't forming the way that they should. So we have a breakdown of the structure of the DNA. Proteins are very similar to this. They're amino acid based, but it's a similar concept. So yeah, you can see how dangerous pH shifts can be in biology, just looking at it that way, right? That's a huge deal when we get into talking about the next unit. Next unit is like all what comes from this, okay? But yeah, so that is the concept of why hydrogen bonds are important and why pH is important, okay? And how pH can affect those things. Okay, whatever with reactants and products. By now, you should know what those are. The things reacting and the things that are made are products. Solutions. These are terms that people are not always familiar with that I will probably never use in my class, but we have the solvent, and I have to start with that one because I always forget which one is which. So, so I think about what is dissolving the thing. So that's the solvent with the V, okay? So it's doing the dissolving. The thing that's breaking down is the solute. So you can think of it as giving a solute as it goes down to be dissolved, and it's not going to be itself anymore, right? We know what a concentration is. Come on now. We know why water is a great solvent. It's because of the polarity of it. And all these polar molecules that you're trying to dissolve, like sodium and chloride, do great with that polarity. But then what doesn't do good with that polarity? Things like oil and wax and fats, things like that. Why? Because they are hydrophobic, water fearing. What makes them that way? I'm telling you about how all these guys are hogging on to electrons and causing all this polarity, especially oxygen. Oh my gosh. So oxygen's hogging on to that Barbie the most, and that hydrogen's got that positive charge to it because of that. And everything that you're associating with also is doing crap like that. Other polar molecules, they're creating those negative and positive polar charges. They mix together really well because they can mix and match their little polarities. They're hydrophilic, they're water loving. Everybody's getting along. It's great. We all have charges. Yes. But when you don't have a charge, you have nothing to share with the water. You can't share anything with the negative oxygen, and you can't share anything with the positive hydrogen. When does this happen? We see this a lot in nice, stable, happy carbon. When carbon binds with hydrogen, it really is sharing the Barbie equally with the hydrogen. Nobody has a charge. That's why it's one of the most stable molecules and why life is built around it. So that aspect of it, it's stability. And what creates an organic molecule, that sharing of the Barbie between the carbon and the hydrogen. Now, hydrogen is key. It's not an organic molecule, it's just carbon with oxygen or something. That doesn't make organic. Organic is when you have the hydrogen backbone in there because we have that equal sharing of the electrons going on. Okay, so they don't get a polarity because of that, because we're being nice and equal and we're sharing. All the mean people out there creating, and mean old big sisters and the little sisters who are crying that want to hang out, like creating this big drama with one another. And then here come these happy little siblings that are sharing equally, and they're just like, we don't want anything to do with that. We're good. Like, we don't need that drama. Think of it that way, right? So that's hydrophobic. They don't want to have to do it with any of that drama of the polarity. Okay. So that's going to be the happy carbons and hydrogens equally sharing, no polarity, no charge. They're just chill. Hydrophobic, fats, oils, lipids, waxes, the obvious stuff. If it didn't mix with water, that's what we're talking about. All right, then we have the amphipathic molecules. Amphipathic, amphi always means both. If you are ever wondering in this class, amphi is going to come up a lot as a prefix in a lot of things, like an amphibian. If that helps you remember. Amphibians can do both land and water, right? So both. So same thing with these molecules. So amphipathic molecules, you can have something like with a charged head. Now, forgive me because I don't know how many electrons the freaking phosphate can have. I don't know how many freaking double bonds there are. Don't judge, please. Here's the oxygen. Here's some negative. I don't know. And then here's carbons. And then H. Not quite this pretty, of course, but it's really pretty. It's not quite this simple, but imagine that you have this charged phosphate group head. Now, it's not the phosphorus that's charged. It's the oxygen. It's supposed to be oxygen. Okay. It's got all these oxygens. I think there's another O one here that I left out. Who cares? But I'm gonna make you guys draw it. So, I mean, I can't draw it. Why would I make you guys draw it? Anyways, that phosphate, we know we have a bunch of oxygens around there. I know that. And that oxygens are always going to have their negative, electronegative BS going on. So super negative going on here. And then this tail down here, those happy carbons and hydrogens. So these are non-polar. So no charge down here, and these are polar up here, obviously. So what does this represent? You guys know? It's a phospholipid. You have the charged head and the carbon hydrogen tail. Phospholipid. It's one word. I just can't get it. So those charged heads and the non-charged tails. The heads, because they're polar, water loving, hydrophilic, right? Like Philadelphia, city of brotherly love. That's where I'm graduate school. Then we have the tail down here. This is the non-polar, the hydrophobic portion. This is why it's empty. It's not both going on. So these charged things don't get along well with the non-charged things, and the non-charged things sure as heck don't want to have anything to do with water because there's too much drama. Always starting stuff, water. So the heads will stick together because they're charged, and then they can hang out with the water outside of them and all of that. And this is why you see this development of like head sticking in. Now that one line can create a big circle and protect the tails inside that way. That's from my cell. Or you can have pollen go on. Tails on the other side. And this will go on around in one big old fat circle around yourself. This is a lipid membrane. It's a lipid bilayer. Bi being two layers. They've got layers of these phospholipids sitting up against one another. And the tail's being protected on the inside by that. So this is a weird situation. You have H2O on the outside, H2O on the inside. H2O don't want to go in here, and the in here doesn't want to have H2O in there. That is what keeps your cell intact. That composition right there of those antipathic molecules. This is why nothing can get in and out of your cell unless you have like channels in your membrane to specifically let things in and out. It's an amazing way to control what can get in and out. There's a water can go across, it can stick in the middle, but across and like what we call that. You guys remember what that's called when it goes across the membrane? Osmosis. That's don't worry. Don't worry, it's coming. Chapter 9. Okay. But you guys get it, yeah? So those amphipathic molecules, because of the chemistry of the charged and the non-charged, the hydrophilic and the hydrophobic, the fact that the hydrophilic, water loving, they like the drama. The hydrophobic, water hating, they don't want to have to do with water's drama. That whole thing defines that entire lipid bilayer that goes around any cell ever. Okay? That's pretty cool stuff. That's the power of an amphipathic molecule. We did all of this. Yeah. We did even this. We did even this. Cool. Carbon's great. It does nice stable bonds. Here's some side groups that you don't really, I'm not going to test you specifically on the side groups. I will bring them up occasionally, and it's nice to understand what I'm talking about, but I don't actually expect you to. Okay. All right. That's our first questions. That's our first questions, y'all, and I'm almost done with class. So let's really quick go through the questions. Everybody stand up, everybody stand up, everybody stand up. Well, luckily we've actually covered most of what we're talking about, so I just like to tell it as a big story. Okay, what is a covalent bond? Some of the stuff, I didn't write that one on the board, but some of the stuff might be on the board, just FYI, you're allowed to look at the board, okay? In fact, let me preface this. If I ever give you guys an exam and I have stuff like this written up on the board and I didn't notice it and you guys started on the exam, fair game. Okay, that's my fault. So, all right, so here we have Rebecca. Well, that's Becky. Yeah, covalent bond. Exactly. Good job. So that's a 0.4 delta. What's racking them up? I don't know why I have to pick a delta. All right, ionic bonds. It's a delta. I'm not doing it on purpose, I swear. Jennifer. Transferring, right. So yeah, the sharing will be that covalent, and the other one, the transfer, completely stealing it away because we're super greedy and running off with the Barbie. That's ionic. Remember, ions are just going to be separated from each other because of that. All right. Brief general concept of a hydrogen bond. Logan. Yes. Yeah, it's based on the charge, right? There's no like electron exchange in that bond itself. It's only based on the polarity of those molecules. So I do want you guys to remember that. All right, so that's a point for Foxtrot. All right, this is Charlie. We have Madison. What types of molecules tend to be hydrophobic? Water theory. Madison here? No? Okay. Cool. All right, next, Bravo, Aubrey. Aubrey didn't make it either. All right, I know this person's in here. A point one. We'll have some people that aren't in somewhere. Krista, this one, Bravo.
That's in the blues.
Right. So the things that don't have a charge, that's like the broad way you could look at it, but that's was definitely fall into that category. Good job. And our last one is going to be, what is pH measuring? What's it actually measuring? Claritza. Exactly. Good job, guys. So that's it. You guys can sit down. We probably won't have any more of the rest of the classes I have. We're only halfway through this. We'll only get through what we have to get through until the class is over. I won't push you guys after. We talked about lipids already and how they're made up of those fatty chains, the CHs. That's the whole aspect of a lipid that makes it what it is. Nucleic acids, we just introduced those. Yeah. But these are our biological macromolecules. They're made up of units. What's the unit for a nucleic acid? That's going to be the nucleotide. What is the unit for, I don't even know what to say about the fats. The fats like kind of depends on what you're talking about, but what's the unit for a carbohydrate? Sugars, individual ones, yeah, saccharides, and that sort of stuff. So proteins are amino acids. So that's basically what we're looking at, are these big molecules made up of these building blocks. Like I said, carbohydrates are made of sugars. They can be linked together. I'm never going to make you draw them. I'm not going to make you tell them apart, chemically speaking. I just want you to be aware that they're sugars. Okay, cool, look at them. All right, next we have the lipids. We really already talked about this. It's the hydrophobic aspect of these that make them lipids. So we have triglycerides that have a glycerol group that has an oxygen in it, so it's slightly charged. And then we have the 3 fatty chains coming off of it. That's why it's called tri. They can be saturated or unsaturated. Saturated means it's saturated with hydrogens, so there's no double bonds. Unsaturated means there's some double bonds. That's what gives it those kinks. All right, the unsaturated fatty acids don't stick together. They're better for your body than the saturated ones because they have that double bond that's causing you to use more energy to break that up, to deal with it, and all this. So unsaturated is a little bit better for your body. We talked about a phospholipid, man. We don't need to go over it again, right? Here's membranes. There can be other lipids in the membranes that can stabilize the shape, like cholesterol. Our cells, their shape is determined by the stability provided to them by cholesterol. Cholesterol is a lipid. Okay? Steroids also, like you could say cholesterol, sterol is a steroid. Any other steroids, that they would all fall into that same category. Waxes in general also do. That tuberculosis that we were talking about, that acid-fast stain, it stains the waxy coating on mycobacterium, and they're unique. They have wax called mycolic acid, and that wax is waxy. Okay. So we'll talk about that more when we get into chapter 4. But and proteins, all of your genes code for proteins. So your genes, you'll have a set of 3, like A, T, C, or whatever, and that'll code for, you know, an amino acid. And then those amino acids are going to link together. Ugh. I can't tell the whole chapter on this one board, but I'm going to try. So we have amino acid, which has a carbon with R group on it. There's a story about this. So when I took, what is it called, cell molecular biology in my undergrad here, I was double majoring in microbiology and biochem, and I ended up not getting the microbiology major. So one of those situations, they had like the class that was only offered like certain semesters, and my advisor didn't tell me. And she's like, "We could stay," and I've already had like 145 hours. I'm like, "No, bye!" Like, no, I'll just take my biochem degree and go, man. So, anyways, I had to take this because I thought I was still, you know, getting that degree. And it's basically watered down biochem. And so I already knew a lot of the stuff. The first exam that came around, the guy from the first test was like draw 2 amino acid points together. And like R for the R groups. So I did that, but I drew them with this on this side and that on the other side. Now, if you are a human being or at all familiar with chemistry, then you understand that that's the same thing. You can look at a chemical from the other side and it's still a chemical, right? He kind of wrong. And I got it up to him, the only question I got wrong on that test. So of course I was going to find it. I went up to him and I was just like, I'm pretty sure, you're not going to get about it, but I'm pretty sure I got this right. And I was trying to like kind of explain why it was right and everything. He's like, oh, and he literally stood up in front of everybody and said, she thinks she knows better than me. She thinks she thinks she knows better than the professor. And it was not a class this size. It was like 100 people in the class. And I said, oh, that's how. I'm sure you guys can imagine I have a back wound. So I took it to the head of the chemistry department at OSU Solar. And he, of course, he was like, well, the stereophonics, the atoms would be coming out this way. And I'm like, it's right, though. He's like, yeah, of course it's right. And I'm like, hey, can you email him? I had him email. I had the head of the biochemistry department email. So I came into class the next day, and I didn't push it. But he like called me up after class. And he's like, hey, I'm sorry, you were right about this. And that's pretty much what it was. But he didn't say it in front of the whole class. Guys, if you think that you're wrong about something, if you think that you're right and I am wrong about something, and you can argue and support yourself, you might still be wrong, right? You might just have the concept wrong or something. But we can at least talk it through. If I maybe wrote the question wrong, or like I wrote it weird, so that your answer actually was right also, other than the one that I did. Whatever it is, if you think that you're right and you can tell me why you're right, you really are right, I will admit it in front of the whole class. I'm not one of those people that's going to be like, sit down, you got your point off, take it. I'm not ever going to do that. So please. Okay? I'm here to help you guys learn. The grades are something I have to do. That's part of the deal. Okay? To show me that you're learning. Anyways, this is amino acid. So this is an amino acid that our group is going to determine what amino acid it is. Whether we are talking about arginine or glutamine or proline or tyrosine or whatever of the 20 different amino acids that we have that you don't have to memorize. You're welcome. So what they might be non-polar in that R group. They might be polar in that R group. That's kind of starting to paint a picture of why it matters what amino acids are in what order to make whatever protein, right? Okay, so that leads us, I just want to get at least done with this. I'm almost there. Peptide, we know that's a peptide bond. Polypeptide is a bunch of them linked together. Okay, primary structure is just going to be the amino acid sequence. So whatever, glutamine, arginine, lysine, whatever, one after another. Think of it as a long like string of ribbon. Okay, that's it. That's primary. Secondary structure. When those R groups that are next to each other. Imagine that you're polar and you're next to a guy that's also polar. So you might get along really well. You're going to get along well with the water around you. But what if you're a bunch of hydrophobic guys? You guys might want to be huddling into that ribbon, surrounding yourself so you don't have to interact with the water around you. This can generate structures called alpha helices, from not right. Helices, that's literally what it sounds like, the helix. Or beta sheets. They look like, they're called beta pleated sheets, but they're literally like going back and forth just flat. You don't even know why they form that or anything like that. I'm not going to make you guys understand that. Yes, it's based on the R groups, but that's all you need to know. Okay. So based on that, whether it's polar, whether it's non-polar, all of those things determines whether we're going to make next to us these alpha helices and beta sheets. Well, now we've interacted with what's next to us. Maybe we've twisted our ribbon. Maybe we've pleated some of our ribbon, right? But now, because we've done those things, we're brought closer to other parts of the whole ribbon. So then we can start interacting with the R groups across the way. We can make disulfide bonds, which are covalent bonds, or other hydrogen bonds with other R groups across the way. And once that whole ribbon is happy with how it's folded because of those R groups, then you have your tertiary structure. And I'm sure you're looking at a quaternary though. What is that? Well, proteins aren't just one ribbon. Okay, it's not one polypeptide. They're typically multiple. So think of like one ribbon that's all tied up pretty and nice and it's finished in its tertiary structure, and another one coming together, and then multiple can come together to make a functional enzyme or whatever that protein's going to do. Okay. All right, we did enzymes. I talked about that. We'll talk about antibodies in chapter 16. I'm not going to talk about it now. Denaturing has to do with breaking up those hydrogen bonds, either with heat or at pH, basically. We kind of already talked about that, right, with the DNA. DNA, deoxyribonucleic acid, and then RNA is ribonucleic acid. Deoxyribose and ribose are pentoses. They are 5 carbon sugars. There you go, they're 5 carbon sugars. They're just different from one each other from one OH group on there hanging out. So the sugars really, sorry, I'm just getting my emails. I just forgot that I had this on. Pentose, 5 carbon sugar, phosphate. All right, so we've got that phosphate giving it a charge. We'll talk about the structure that creates for the DNA whenever we get into chapter 6, but it's going to be charged, right? It's got a phosphate in there. We already know that that's danger, so it's coming. So sugars and phosphates on the backbone, and then that's what makes us up the sides of the ladder. What makes the rungs of the ladder, the ACGT crap, those are determined by the nitrogenous bases on that. Don't care about that. Don't care about purines and pyrimidines, okay? I never learned the difference, so no. I have a biochem degree, so that should say something. All right, we talked about DNA, RNA. We'll talk about the functions of these in chapter 6. We don't need to worry about that now. ATP, you guys remember that one? Yeah, so this is our energy molecule. This is our last slide, by the way. Our energy molecule, ATP, adenosine triphosphate. It actually tells you what it's made of right up front if you know how to read it right. We know triphosphate means 3 phosphates. We already said phosphates are danger. They got so much negative charge going on. It's nuts. So adenosine, so we have 3 of those phosphates. Adenosine means adenine with a sugar, and it's ribose. Okay, that's our sugar. So we have adenine, which is like the A in DNA. We have the sugar, ribose, and we have the 3 phosphates. Those negatives next to negatives next to negatives. Well, I told you H is like to associate with O's because the H's are positive and the O's are negative. Did I ever show you guys that the negatives want to get with the negatives? No, because they don't want to. So that's like North Pole to North Pole or South Pole to South Pole. They're going to repel one another. The fact that we have covalently bonded and forced them to share their electrons with one another, and they're repelling each other constantly. They don't want to be there. When you break off 1 or 2 of those phosphate groups, releases a ton of energy. That is potential energy. Same as if I hold this marker up here, potential energy. You guys know there's energy here, right? Here, I'll show you. The energy was just released. That's what's being held in those bonds. So we can break those bonds and release that energy. And they won't just let the energy be broken. They will use that in the cell to help do other things, building molecules, stuff like that. You know how when you build things, you have to put energy in. When you tear them down, you get energy out. So if you're going to be building up new things, you've got new cells, you got to make. You fell and you scraped your knee and you need to make new cells. Well, you've got all these molecules you got to build. You got to make new DNA. You got to make new proteins. You got to make a new membrane. You got to do all this stuff. You better get some energy together. So you're going to constantly need energy. We'll learn about ATP more in chapter 10, but I'm going to let you go, I promise, with one last little tidbit of information. If you have not taken physiology, then you are going to love learning about metabolism. It is my favorite thing to teach. Aerobic respiration, it's like mind-blowing. So let me put it this way. We know we breathe in oxygen. We'll go into details later, don't worry. When you breathe in oxygen, oxygen that you're breathing in, it only has one purpose. Only one, I want to be clear about this. The only purpose that the oxygen that you breathe in is to help maintain a gradient of positive hydrogens outside of a membrane versus inside. And if you cannot maintain that gradient, then those positives don't want to come back in to equal out, right? They have a whole pile of stuff outside and not anything on the inside. So constantly the outside wants to come in. That's diffusion. It's, wants to be equal, equilibrium, all of that. Well, if you let it build up and you let those H's come in and they're equal on both sides, you cannot make energy anymore. What's that energy? ATP. How serious are we talking about here? If I suffocate you, how quick do you die? Very quickly. Why? Because you don't have oxygen, but why? Because you ran out of ATP. No shit, that's why. 100. Y'all, I just brought it around for you. So That's what I'm talking about. But we'll talk about that in more detail in chapter 10. We're not there yet. So you guys are free to go. If you have any questions, of course, come up here and ask me. I'll get to you if I can. Email me. Remember, lab coats and closed-toed shoes on Tuesday. I don't know where it went. Now I've got to erase this. Now I'm going to have to redo this all tonight. Yeah, I would go like the list that's like you're on the site. There are other ones that types of, but I would just like to go list or pictures or whatever it is. I don't need details, like super detail, like methylation curves and you know, tell me about how you know the mitochondria make it. That's really great. Yes, yes, yes. Yes. So if you can. I think you could usually. Feels ridiculous because I'm like I'm gonna do these groups and whatever. And probably are. I checked Oh, yeah. I imagine that's it. Sorry. Now I'm trying to make it as hard as possible.